Also, you are about to see data that has not been published yet, so I will ask you to please don't share graph pictures or any data with anyone without the approval of the authors. So, Dr. Mortreux, you can share your screen.
Okay. So just let me know if you can see that.
Yep, all good.
Okay, wonderful. So hi, everyone. Thank you so much, Margot, for the introduction. So today we're going to discuss what can be expected from hypogravity environments, and we will see if sex would matter for musculoskeletal health on the Moon and Mars. So I just wanted to do a quick background for me. I did my postdoctoral training from 2016 to 2019 at Harvard Medical School, and then I became an instructor in neurology until 2022. I'm actually leaving Harvard now to become an assistant professor at the University of Rhode Island, where I will start late August, and I will start the metabolism and muscle biology lab. So I have worked on NASA-funded studies since 2016, and I am currently the staff scientist or co-investigator on 3 NASA-funded grants, including one with a bed rest study in humans. And I have 3 more NASA grants currently under review, either as a PI, as a co-I, or as a consultant. So I just wanted to give you a quick outline of the talk today. We're first going to focus on the musculoskeletal response to microgravity. We're then going to shift gears to look at the response to partial gravity, especially Martian gravity. And then I really want to take a look at the musculoskeletal adaptations during gravity shifts, because this is very novel and it's not done yet by any people. So just a quick introduction. As you probably all know, mechanical loading or gravity is the most important stimulus for the maintenance of the musculoskeletal system. And the adaptation to microgravity is not very well understood thanks to spaceflight, but also ground-based analog models. So as of October 2021, and I'm sorry, I don't have the most recent numbers, 579 people from 41 countries have gone to space since 1961. And while some of those flights are just a few minutes, like if you think of Bezos, Branson, or even Alan Shepard, the record is held by Gennady Padalka, who spent 878 days in space, which means that we have now a good understanding of how short and midterm adaptations to microgravity for the human body. And as I mentioned, we also use ground-based analog models. So on Earth, we use a head-down bed rest with a tilt at 6 degrees, which allows us to simulate sweatlessness, but also the syphilic fluid shifts that is experienced on the space station. The good thing about ground-based analog is that you can actually include a wider range of participants. So for example, you can look at different ages, but also include more women, which is going to be kind of the topic today. Another possibility is to add countermeasures, which is sometimes complicated to do in space due to limited crew time, due to the regulation, or just even having the equipment on the space station to perform those countermeasures. So beyond low Earth orbit, we actually find multiple targets of very high interest, most notably the Moon and Mars that I'm sure you can recognize here. And in both cases, the astronauts would have to evolve in some partial gravity. So we're looking at 0.16 G on the Moon and 0.38 G on Mars. But what experience do we really have with partial gravity? So we know that the Apollo missions allowed some lunar EVA of a few hours with some additional time spent on the surface, but inside the lunar module. And while several rovers like Perseverance here have been sent to Mars, humans have yet to reach and explore it, which means that we don't exactly know what to expect in these partial gravity environments. So I just want to point sex difference here because you're going to see it throughout my talk. As of March 2022, only 75 women have flown in space. And that actually includes the payload specialists, so the scientists, and it represents less than 12% of the number of people who have made it. And if you're too young to remember Apollo, it consisted of 12 men and 0 women who were sent to the moon and the lunar surface. So we don't exactly have parity there. But the space agency have been trying to do better, and you can see it with NASA and the selections they did for the Artemis team, which will send the humans back to the moon and prepare for Mars, where you have an absolute parity with 9 women and 9 men astronauts. The problem is that even in research studies, females or women represent only a fraction of the data when they even exist. And that creates a very important and dangerous gap in knowledge because we just assume that women will react the same way as the men. And I'm about to show it's not as easy as that. So since we're lacking direct models for some of the studies, there was really a pressing need to establish rodent analogs. So what you can see here, for example, is a drawing of a normally loaded rat, which I will refer to as NL, which is used as a 1G control, and my work focuses on those rodent analogs. In the 1970s, Emily Murray Holton created the hind limb suspension model, or hind limb, or HLS here, where you can see the rat is suspended either by the tail or by a pelvic harness to induce weightlessness on the hind limbs only, and it simulates microgravity, so 0G. And the problem was, during my postdoc, I was tasked to see what is the effect of partial gravity, but there was no model for rats for me to do that. So I created it. And it's called the partial weight-bearing model, or PWB, where you can see that the front limbs of my animals are supported by a jacket, whereas the hind limbs are supported by the same pelvic harness that you can find in the hind limb suspension model. And this allowed me to simulate partial gravity by changing the length of the chain here and reducing the weight the animal is able to put. And I can basically mimic anything between 0 and 1G, but I usually focus on the moon and Mars using 20 and 40% of their weight bearing. So we're first going to start with the musculoskeletal response to microgravity, and I'm going to show that in both male and female. And this work has been published last year already, so you can refer to this paper. And I just want to give you some methods. They are globally applying to every single study I did. So I use outbred Wistar rats because they have genetic variability, which is very similar to the one you can find in humans. They are 14 weeks old at baseline because they are muscularly and skeletally mature at this age. And to induce a very severe deconditioning, we put them in hind limb suspension for 14 days, so the equivalent of 0G. And while we do some longitudinal in vivo assessments of muscle function and bone mineral density, we also complete that with a lot of ex vivo analysis. So in microgravity, you expect to see some weight loss due to metabolic wasting, but also the loss of lean mass. We all know that we're going to lose muscle and bone when we are in space, and those weigh a lot. And the studies on the ISS shows that in humans, you have a weight loss of around 2.4% for every 100 days you spend over there. So if you look at my animals, here the males are in black and the females are in gray. You can see that the males actually lose some weight during the hind limb suspension, and it's significant after 14 days. But the females did not. So they do lose a tiny bit of weight, but it's never significant. And during the second week, you can see they actually are going up. So grip strength is just literally the strength of your hind limbs to be able to grip. It's also a test we do in human. And here you see the hind limb grip force in healthy males go slightly up over time, which is absolutely normal. But let's look at the dashed line here for the males in hind limb suspension. What you can see is after 1 week, we have a decrease by about 30% of their grip strength. And by 2 weeks of hind limb suspension, you have a decrease of 60%, which is almost perfectly linear. Now, if you look at the females, you can see a more moderate decline in 1 week because they reach approximately 25% of their baseline, but it's really restricted to this 1 week. During the second week, they are stable and they end up at 24.3% of their baseline at the end of the experiment. So we also look at force production because the force you can apply or you can grip is a bit different than what is the maximal force your muscle are physiologically able to produce. And so for this, we use a foot plate and we do a stimulation of the nerve to induce a tetanic contraction. And what you can see here is the force production during a dorsiflexion, which is when you dorse your foot. And it mostly involves the tibialis anterior and the EDL muscle. What we can see here is that in 1G males, the controls, it increases over time, which is what we are expecting to see. But if you look at the hind limb suspended males, you have a sharp decline during the first week. If now you look at the gray bars, the females, you can say it's very stable regardless of the gravity we put the animals in. And so I didn't actually put the picture for the force production for the plantar flexion, which this time involves the gastrocnemius and soleus muscles, but it does fail to increase in the male, and it remains very stable in the female as well. So we were wondering, okay, if the force is different, let's take a look at the muscle. Well, when you go to space, you expect muscle atrophy, and this is something we can see quite clearly here in the gastrocnemius, where both sexes lose approximately 20% of their muscle mass. But what's really interesting and really surprising is that if you look at the soleus, which is the anti-gravity muscle because it's weight-bearing, and it's the one that responds the most quickly to unloading or mechanical reloading, you can see that the males have a greater atrophy in this muscle, as well as in tibialis anterior, which is the biggest muscle responsible for dorsiflexion. We see this trend once again in the EDL, but it does not reach significance. So females have better force, but they also have like smaller atrophy or less atrophy. So now we want to take a look at the bones. And you expect bone density to decline. And in space, for example, in astronauts, we see a bone loss of 1 to 2% per month, which is twice the rate of postmenopausal women. And what you can see here using PQCT, which is peripheral quantitative computed tomography, we can look at bone mineral density over time. And after just 2 weeks of Hahnemann suspension, you can see that females lose approximately 5%, but this remains within the variation we actually see in the healthy controls. If you look now at the males, we do have a 20% bone loss after just 2 weeks of simulated microgravity. This is 4 times more than what we saw in the females. And if you're interested in this and you are going to do some literature, also be mindful that very interestingly, females have a higher bone mineral density than males, even if you just look at the raw value. So we really wondered what can be the reason for the female's advantage in this condition, which is simulated microgravity. And what we did is we looked at vaginal lavage to check the estrous cycle of the females. And by doing that every day, we were able to see some correlations between estrous cycle and grip strength and estrous cycle and bone mineral density, which means that the animals who maintain their estrous cycle were the one with the biggest strength and greater bone mineral density. And the one who either became acyclic or just spent a lot more time in those very low hormonal states were the one who showed the greatest decline in those parameters. So it seems that the preservation of the estrous cycle may be able to mitigate deconditioning, or at least is significantly correlated with this deconditioning. And if you look at the paper I referenced here by the postdoc in the laboratory, Megan Rosa Caldwell, you can see that it's also something we observed in cancer, where when you induce cancer in animals, you can see that the females who maintain their estrous cycle never develop cancer cachexia, whereas the females who lost their estrous cycle do. So just to summarize, in microgravity or simulated microgravity, we see that females better maintain their body weight, they better maintain grip strength, they maintain their force production, they better preserve muscles from atrophy, and they better preserve bone mineral density. And we do know that at least in part, the musculoskeletal deconditioning is linked to the estrous cycle maintenance. Now let's shift gears and look at the musculoskeletal response to partial gravity in males and females once again. And this is actually going to be based on 2 different papers that we published looking at Martian gravity analogs. So we use this partial weight bearing 40, so 40% of weight bearing to mimic the 0.38 Gs that you would have on Mars. And those are 2 exactly identical protocols that we did just in males and females. So similarly to what we just saw in microgravity, you can see weight loss in the males here in blue, whereas the females do not lose weight at all. But we all know females are better at conserving energy and especially body mass, so it was not that surprising. We then look at grip force. So first, let's focus on the males. At 1G, you can see as before, you have this increase over time, which is normal. But if you look at the males in Martian gravity, you can see a sharp decline. I think it's around 30% during just the first week. And it tends to go up after that. It's actually something we see quite often in partial gravity. But if you now look at the females, both groups remain very stable over time. So we then looked at the muscle, and for example, we looked at the gastrocnemius, which is not the first muscle to atrophy, but it's still a very important calf muscle. And we see here that males experience an 11% reduction in muscle mass. We then look at the females, and we didn't see anything. So we were wondering, okay, maybe the mass is the same, but maybe the myofiber size is going to be impacted. Well, it was not. So the males had this very large reduction in type 2 fibers and average fiber size. It was a reduction of around 20%. And as you can see, the females, we did not see any significant differences. But as I just told you, the soleus is, the most responsive muscle. So we looked at that, of course. And you can see this very dramatic atrophy after just 2 weeks of Martian gravity in the males. But once again, if you look at the females, we don't see anything. So we went and did the histology, and we found this time a 28% reduction of myofiber size in the males only. No differences were found in the female animals. So just to summarize here, in a Martian analog using partial weight bearing, we find that females fully maintain body weight, they fully maintain their grip strength, fully maintain muscle mass, fully maintain myofiber size, while males show significant declines in all of these parameters. And so far, it may seem that females are much better equipped to handle reduced gravity than males. And when I started my study 6 years ago, we initially only had males, and we were really looking at the impact of partial gravity. And it's something that we didn't do in females at all. It was not part of the grant. We didn't have the money or time to do that. So I just really want to show you some results from this study so you better understand what partial gravity actually does to a healthy animal. And this first grant I worked on is largely summarized in a few papers, but the two main ones are one in muscle and one in bone, looking at the longitudinal time course of what's happening when you are reducing mechanical loading. So the main questions we had for the study was that, is deconditioning linear along the gravity spectrum? Like, we know what's happening in 1G, we know what's happening in 0G. Is it linear between those 2 points or not? The second question was, does it occur at a steady rate throughout the experiments? Or is it like really important in the first week? Is it really important at the end of the months? Like, we really didn't know the answer to this question. Another very important question for us was, does it ever plateau? We know a lot of systems adapt eventually to microgravity. Is it true also for bone and muscle? And the final question we wanted to ask was really, is there a safe threshold for weight bearings that does not result in deconditioning? So for example, for NASA to be able to say, well, if you keep your astronauts at 0.7 G or 0.5 G, they will be fine. So this is what we were really trying to see. And we use 4 different gravity levels, 1G, 0.7G, 0.4G, and 0.2G to mimic the moon, Mars, Earth, and 1 moderate gravity. And why are those questions important? What is because if you look at this graph here, where you can see the adaptation of different systems to space flight, you can see first that muscle is not necessarily there. You just have lean body mass, which seems to plateau eventually. But if you look at bone here in very light blue, you can see it's actually linear and never plateaus. So we first started with grip strength, just like you saw before. And as you saw before as well, you can see the controls increase their grip strength over time as they age. It's normal. Rats are actually constantly growing. This is exactly what you would expect. But what you can find is that grip force declines very quickly. So you can see here, after 1 week, all of our groups in partial weight bearing had this significant decrease in grip strength, and we're looking at between 20 and 30%. What we found is that it also continues over 28 days. So it does not stop. And the question we had was, is there any protection from this moderate gravity of 0.7 G? And while you may think this is like a small reduction, this is a reduction compared to day 0. But if you compare to the actual control, we are looking at a 60% difference in those values. So there is no protection at all from partial weight bearing 70 for grip strength. And as we did before, we used the foot plate to see if the muscle could physically produce more or less force over time. So we look at the difference between a baseline and at the end of the study, which was 28 days. And what you can see here is exactly the same profile, with the 1G animals being able to produce more strength when we apply a current, and the partial weight bearing animals being able to display less and less strength over time. And what you can see here is this visible dose dependence for force production. Of course, we completed that with muscle mass and histology. And as you can see here, while it declines, you can see the group at 1G, 0.7, 0.4, and 0.2. It's not necessarily always linear, though some appear extremely linear. 0.2 is always kind of the odd group we have, and we think it might be just because the variation are bigger when you're at 0.2G than when you're at 0.4, 0.7. But what's interesting is, if you can notice here at day 7, the CSA for the 1G animal is around just lower than 3,000 micrometers square. And after 28 days, because the animals are always growing and gaining weight, they're actually bigger. So you might think, oh, but the myofibers are bigger after 28 days and 7, so surely it plateaus or it begins to recover. But if you look at the difference between the 1G group and the other, you will see that it actually increases over time. So the growth rate of the animal is not enough to mitigate the atrophy. And we also looked at bone, of course, and you can see here bone mineral density using PQST, which is just the same thing I showed you earlier. And once again, after just 1 week, you can see this dose-dependent decline. And for the bone, we're really looking at something extremely linear. And you have this clear dose response, not only for the first week, but also on week 2 and week 4. And once again, no plateau. So we end at approximately 30 to 40% bone loss after a month. And once again, if you pay attention here to the pound side, 0.7G was not able to prevent this bone loss from happening. So bone homeostasis is a balance between bone formation with osteoblast and bone resorption with osteoclast. So we wanted to know which one was impacted that threw this balance off. And here you can see osteoblast surface, which is labeled with those yellow triangles. And you can see many of them in the 1G animals, less of them in the 0.7, just a few of them in the 0.4, and none of them actually in the 0.2. So what we showed, and you can find in the paper, is that the bone loss was mediated through reduced bone formation, but it actually was not due to an increase in bone resorption, which is pretty interesting. So now let's go to the big part of the study, which is the musculoskeletal adaptation to gravity shifts. And it's going to be in males and females, but not for every single condition, so I apologize for that. So the first thing I want to talk to you about is I tried in the lab to mimic what will happen if we actually implement artificial gravity on the gateway. So I'm sure you're all very familiar with the space station and what the gateway is, but the gateway is just going to be a station orbiting the moon, a bit like ISS is orbiting Earth, which will allow for months-long mission on the lunar surface. And this is what we are currently trying to build with Artemis. And we're going to use that as a base to go and then explore Mars. But let's pretend we have artificial gravity on the gateway as a countermeasure. The trip will actually not just involve artificial gravity. So first, we will have to leave Earth at 1G and go into the Orion capsule, go to the gateway. So we're going to spend time in microgravity. Then while the gateway is orbiting, we may be into some artificial gravity. Then we will have to do the trip back. So once again, being exposed to microgravity before landing on Earth and recovering there. So because this might be a valid countermeasure, we really wanted to see what would be the best artificial gravity to have aboard to make sure that the astronauts return healthy and safe. So this is a graph actually from the grant that summarizes the protocol we're using. And I'm just going to walk you through it because I know it's a lot. So we first mimic the trip to the gateway by putting the animals for 2 days in helium suspension to mimic microgravity. Then for 14 days, we're going to have some artificial gravity protocols. So some animals will be just spending 2 weeks at 0.2 G. Some will be just spending 2 weeks at 0.4 G. Some will be just spending 2 weeks at 0.7 G. And one very special group, which we will see later on during the talk, is going to have a fractional reloading. So we're going to slowly increase mechanical loading over 2 weeks from 0 to 0.7 G, just to see if that has an impact. And after that, we do the trip back. So once again, 2 more days of hind limb suspension. And then we're going to see how the animals readapt to Earth's gravity. So we wanted to look at 4 hours, 24 hours, and 7 days. And this is just because we know recovery might be complicated and coupled with some injury. So we really wanted to look at both acute and midterm effect. And so, as I said, the caveat for this studies is that it's males only for this one. And those results are unpublished and not always fully analyzed, so they're not fully formatted with all the stats available for it. So I apologize for this. So what we can see here is after 2 days only of hind limb suspension, you can see a decrease of around 20% of grip strength already. But then if you look at the period of partial weight bearing or artificial gravity, you can see that basically in all group, we see some stability in here. No recovery, no worsening. The only thing we see is maybe the 0.2 G group has a trend downwards. What's really interesting, and I was actually very surprised by, is that the 2 days for the return trip lead to an additional 20% decline in grip strength, which I thought it would be a lot less because the animals are already disused. But no, it's really fully additive. What we can see from this graph also is just 24 hours of recovery at 1 G has a significant impact and allows animals to recover a lot of this grip strength they have lost. So then we really wanted to put the animal through a fatigue protocol. So the soleus, which is a weight-bearing muscle, is mostly slow twitch fibers that are contracting all day. And what's the problem with astronaut is you have muscle fatigue and weakness. So we really did this fatigue protocol where you have 2 minutes with 1 contraction per second. And here, those graphs are counterintuitive. A high force drop during fatigue means that you're losing more strength during the protocol. So a higher number means that you have a greater fatigue. And what you can see is after 2 days of hind limb suspension, you do have a greater fatigue in every single group. What we can see after that, it's that during partial weight bearing, it actually makes its way down back to almost baseline levels. But if you look at the last part, and so this part is a bit complicated, it contains both the 2 days of hind limb suspension and the 1 day of post-landing recovery at 1 G, you can see that the fatigue increased again. And it's especially true in the animals who went and had 0.4 G of artificial gravity. So that was not something we're necessarily prepared to see. So we looked at a different parameter to see fatigue. This one goes in the normal direction. So lower values are worse and higher values are better. And you can see exactly the same profile. So more fatigue after 2 days of hind limb suspension, recovery during artificial gravity. And then if you look at this 0.4 G group in red, you can see this drop in fatigue resistance after the return and post-landing recovery. So we wondered, why does it happen? And if we can find a difference with this 0.4 G group compared to the other one. And when we look at muscle mass, so either in the gastrocnemius or the soleus muscle, we really didn't see much differences. And we looked at myofiber size in the soleus, and once again, it seemed pretty similar throughout. So we then look at fiber composition, because if you have different fiber types, some that contract slow, some that contract fast, and some that contract that are hybrid, that may explain a different fatigue profile. And what we found here in the soleus is that the partial weight bearing 40 for artificial gravity actually have slightly more type 1 fibers and less hybrid fibers than the other. So the problem is that during this fatigue protocol, we do have the implication of the soleus, but the main contributor is really the gastrocnemius muscle. And so while the difference in fiber composition could explain what we see during fatigue, we would really need to go further and finish the histology on the gastrocnemius muscle to see if we do find the same pattern. So in this context, we did not see a dose-dependent deconditioning due to reduced mechanical loading. And you just saw like 10 minutes ago that 2 weeks of partial weight bearing normally induces this linear muscle and bone deconditioning. So we may wonder, is it just because they all represent somewhat of a mechanical overload because all those animals were first exposed to a 0G analog? What is really concerning for me is that after 2 days of microgravity at the end of the mission, they have the same dramatic impact than the one at the very beginning. And this is something I was really not anticipating. So the next step for us is to investigate how animals recover back at 1G for a longer period of times. As I showed you, some studies will go to 7 days. And maybe this artificial gravity protocol, maybe we'll see one group that actually show a quicker recovery over time. So just as I said before, we had this multi-partial weight-bearing group, which was fractional reloading. So instead of being exposed to artificial gravity for 14 days with a one set of gravity, they went 4 days at 0.2G, then they spent 5 days at 0.4G, and then 4 days at 0.7G. And what I thought was that the slow increase in mechanical loading will be beneficial for muscle health because it will induce recovery, but not injure the muscle too much, because I thought, baby steps might be just the way to go here. And so if you look at the grip strengths, which we just saw for the other group, it's actually the only one that shows after this 20% drop here, that shows a trend upward during the artificial gravity protocol. We still see this strong decrease after the 2 days of hind limb suspension at the end of the mission, but we do see a small improvement here. When we looked at fatigue, and this is where I'm actually very happy with this result, you do not observe the increased fatigability during the second period of hind limb suspension and landing. So you still see this fatigability increase after hind limb suspension, going towards recovery during partial weight bearing, but still going towards recovery after the second period of hind limb suspension. So you may wonder, okay, what's the difference? Why are those muscles less fatigable than the ones that just had one level of artificial gravity? So here, I just put the other group for you to compare. You can see that muscle mass is similar. I mean, it's towards the high end of the groups, but we didn't see difference. And when you look at cross-sectional area, it's the same or maybe smaller. As I said, we have not finalized all of the statistical analysis yet, but it looks like there might definitely be some significant difference between the blue and the purple one. So we looked at the fiber type profile, of course, and what we found in the soleus is that the purple group, the fractional reloading group, is actually very similar to the 0.4G group. But if you remember, the 0.4G group is the ones that did worse when it comes to fatigue, and the fractional gravity group is the one that did the best. So it really gives me even more incentive to go and look at this gastrocnemius histology to see if that can explain what we observed during the fatigue protocol. So as we just saw, mechanical reloading can have two effects. It can induce recovery with lean mass gain, and it can represent an additional trauma, especially for the conditioned system that adds a new injury. So what we did is we designed experiments to assess musculoskeletal function after traveling in space and landing on the Moon, Mars, and Earth. And we really wanted to look at both the acute and midterm effects. So for today, and because we only have a few minutes left, I'm just going to focus to the landing on Earth after a long trip in microgravity and looking at what happens after 7 days of recovery on Earth. And for this, of course, I'm looking at males and females this time, not going to disappoint you. And we're going to start with bone. So if you remember, you saw this figure before, but kind of cut at 14 days. We do have this minimal bone loss in the female during hind limb suspension, and this 20% bone loss in the males after just 2 weeks. What's really interesting is that the second part of the study is at 1G. So you would expect animal to recover some bone density because now they have the full mechanical loading required for the maintenance of the system. And you can see that it's true in the females. you do have the stability, but in the males, even after landing, they continue losing bone in a very linear fashion. And just to give you an idea, we're at minus 20% after 2 weeks, and we're at minus 27, 28% after 3 weeks. So it's really almost perfectly linear. And the females really don't. And it's nothing anyone had observed before. Sorry. So this was bone. Let's look at muscle, which is kind of my main interest. And let's look at rear paw grip strengths, which we have seen many times during this talk. And here you can see that during hind limb suspension, you do have a sharp decline. We're talking over 40% right here during the 2 weeks of simulated microgravity. If you look now at the recovery part here, you can see that as the females exhibit a better recovery, and they actually exceed baseline levels, while the males do show a recovery, but it's just not as powerful. So then we looked at force production, and this is where I'm showing dorsiflexion. So remember the tibialis anterior and EDL compartment. And what you can see is that we have very minimal effect during hind limb suspension, but in males, it declines after landing and readjusting to Earth's gravity. But if you look at the females, it increases significantly. And so we looked at fatigue. We're like, okay, the force is one thing. Let's look at the fatigue. And when we look at the fatigue protocol, we didn't see a better resistance in females or in males. We kind of saw the exact same pattern, which means hind limb suspension makes your muscles more fatigable, and recovery seems to improve that to some extent, but there is really, as you see, only a significant effect of time. We really don't have that much of an impact after recovery. So then we look at muscle mass and myofiber cross-sectional area. And what you can notice here is that overall females have a smaller myofibers than males in the soleus muscle. And it's actually only significant for those type 2 fibers, which are representing less than 20% of the soleus in those disused muscle. But what I want you to keep in mind is that is normal rats. So if I take just rats at 1G and we publish on that, the female's fibers are normally 14% smaller than the male fibers, just because of the sex difference and mostly because of the weight difference. But here, when you do the math, you actually see only a 7% change between male and female, which might mean two things. It might mean that overall the females experience less atrophy, or it might mean that they recover more during the reloading period. But what I can tell you is that the difference here between males and females is not as important as it should be. When you look at the myofiber type here, we didn't see any differences, but the one thing you also need to keep in mind is that those muscles are still not looking normal, because in a normal muscle, you would have here 80 to 90% of type 1 fibers, which is not the case here, where they are hovering between 60 and 70. And just to show you what we are using to actually determine fiber type and cross-section area, and I hope you can see, we use WGA that marks the cell borders, and we use that to calculate the cross-section area of every single fiber. And then we have myosin heavy chain type 1 and type 2. And when doing the merge, we can see the fibers, for example, this one, that are co-expressing both type 1 and type 2 myosins that we then label as hybrid fibers. So what we saw is that the adaptation to 1G following microgravity seems to allow for a better recovery in females compared to the males. And despite similar muscle mass and slowly smaller fibers, females actually show more endurance and better force production over time. And the females did not lose bone during or after hind limb suspension, while males lost 10% per week. And remember, I told you in humans, 1 to 2% per month was twice the rate of postmenopausal women. So in conclusion, I just want you to remember a few things. Partial weight bearing impacts healthy and disused animals differently. So in healthy rats, partial weight bearing or partial gravity induces a dose-dependent deconditioning, which is steady, it's linear, and there is no safe threshold, at least up until 0.7 G, to maintain musculoskeletal health. But in disused animals, actual gravity using partial weight bearing represents a fair countermeasure independently of the amount of mechanical loading you're applying. But what we saw is also that a gradual increase in mechanical loading seems to be the most effective strategy to preserve muscle endurance from fatigue and to maybe minimize post-landing injuries just in males. I'm not saying this is going to apply to the female work. And so in micro and partial gravity, females do not respond similarly to males. So we are making this false assumption that all humans are equal. It is not. And in microgravity or in Martian gravity, females experience less or no deconditioning at all. And females also show a better muscle recovery at 1G after high suspension, and you can actually extend that to bone, which is strikingly stable in the females exposed to disuse, but dramatic in the males exposed to disuse and recovering from disuse. And so that really emphasizes the need to conduct more studies in females and in women. And I hope if any of you are involved in lab work, you would just take this into consideration. And maybe the message for NASA is just, we should only send women to space. So I just want to acknowledge the funding agency, mostly NASA and some fellowships that funded my work over the years, the people who have taken part in this and you, and I would take any questions you have.
Well, thank you for this talk. It was really amazing. We have several questions in the chat. I will read them. Okay, so we have some thank yous. So first question, since there are baseline difference between males and female rats, is it possible that there is a regression to the mean affecting the results?
So it's a very important question. We actually look at all of those values, both as raw data and as baseline changes, because as I mentioned, we have this tremendous genetic variability. So unlike mice, we do not expect them to be identical. And we do find the same result regardless of the raw data or percentage change. And it's actually strikingly similar. And if you notice my error bars, they are quite tight. So we are pretty sure that those are really like real effects as opposed to a distortion from where the true mean could be.
Okay, the following question was, do male with low baseline and female with high baseline follow the same pattern as other subjects from the same sex?
Well, I can't tell you if they follow the same pattern. I can tell you even with raw values, we do find that within one sex, they are all fairly similar. Like we don't have any outliers that need to be removed, but we also do make sure of that by using a high N for each group. I usually strive to do 12, even though you saw some data with less than that.
Second question, do you think that differences in perfusion between males and females could be one of the major factors behind the observed differences?
It's a very good question. So I think it depends what you call perfusion. We actually did look at the cardiovascular system to look at blood flow, and we see that there was no impact of the partial weight bearing level, but it's not true if you look at hind suspension, because you do have this cephalic fluid shift. And unfortunately, it's not something we have looked at.
Third question, it is indeed very important to test both male and female subjects, but we have noticed that it's very difficult to recruit females for some experiments, including head-down bed rest. Do you have any advice to help recruiting more female volunteers?
I think that we have a problem with that is probably a problem incentive, and I think it's probably a social problem. Women are still the one taking care of the house and children and the primary care provider for them. And I think we need to change the society as a whole if we want to include them more in something that requires them to stay away from their home and children or husband for months at a time. I think also the fact that women are already so sensitive to osteoporosis. I mean, on Earth, when you look at osteoporosis, you're really looking at postmenopausal women. I think we're just so much more aware that this is a problem, a health issue we're going to encounter later on, that they may be just scared of taking this risk. And so maybe this is something we could definitely look at, especially by using some drug treatments like abaloperitide, for example, but we should definitely strive for parity.
Follow-up question, since the menstrual cycle has an impact on the results, what is the impact of birth control on the results?
That's a very excellent question. I don't know. I haven't done this experiment yet. So we did actually look at the impact of sex hormones during hind limb suspension and Martian gravity analog, but we used a different strain of rats and they were ovarectomized, or the males were castrated. So we just know that the astronauts, most of the women are on birth control pill. And usually just for practicality purposes, we know some of them at least take it continuously so they don't have period. But I think it's probably often a factor that is just not even recorded. And we really need to make sure we record cycles in the females so we can maybe see correlations between what we observe from one study to another, because if you look at the literature, you would find studies saying males lost more, females lost more, and all of them just contradicting each other. And I think if we could normalize with the ester cycle, it would be extremely useful.
Another follow-up question was, what good practice could be used to reduce the bias induced by differences in the menstrual cycle?
So the cleanest way to do that, at least in rat models, is to do an ovarectomy for the animals. So you really don't have the ovaries at all, and you just supplement them with, for example, just estradiol, just progesterone, or we could think of supplementation daily, a bit like we have with birth control in humans, to give that to the animals. So good thing with giving them some estradiol or some chemical agonists is that we can single out some receptors. So we know a lot of work has been done with estrogen receptor alpha, but we may want to look at the effect of the beta separately. So chemical agonist and supplementation might be the way to go here.
Okay. Last question in the chat. What is the impact of your finding regarding the use of artificial gravity as a countermeasure for long duration flights, for example, for going to Mars?
I think that's a very tricky question to answer. So it depends when you're starting to implement this partial gravity or artificial gravity, right? Because if you look at my study, when you just take healthy rats and put them directly, for example, at 0.7 G, you still have this musculoskeletal deconditioning. So if I took my astronauts and just after landing, you put them in 0.7 G, may we minimize the loss they will experience on the trip to Mars? Probably, yes, it's going to be less than what they would experience in microgravity. But if you wait for a few days and they change vessel, or they have a halfway point, which is, I don't know, another planet or another time of the flight where they can safely transition to artificial gravity, will the microgravity period initially be just so much? I can't really tell you. And it's really interesting to see that after, on this gateway paradox, it seems pretty independent of how much gravity we applied. But if you just bring healthy animals in a partial gravity environment, they will lose muscle and bone. I can guarantee that.
Okay. Does anyone have a question? Do you want to speak up? Okay, doesn't look like we have more questions. Well, thank you very much, Dr. Mortreux, for hosting this webinar. And I see you guys soon for the next webinar. Thank you for coming and have a great rest of your day.
Bye, thank you.